There are diseases where you look inside the brain and you see a hole, and it's pretty obvious why that brain is not working properly. But there are other diseases where we don't see anything, and we need to look closer. And one of the reasons might be that there's a circuit dysfunction. Some connections are not right. And until we can figure out how to map connections in the human brain, we basically won't know that. You know, I can remember the first time I held a human brain in my hands back when I was a student. And it was sort of a humbling moment. You know, somebody had given this brain for research after their death. And yet, I'll be honest, it didn't look that impressive. You know, it's about 1.5 liters of sort of, squidgy gray stuff. And yet there's all of that, all of this function in there. And I guess at a similar time, I started to get into looking at the inside of the brain. And that's really where the magic, and actually I'd say also the beauty is. So these individual nerve cells, the neurons. So this is an example of one that's actually involved in controlling movement. So they're specialized cells. They have input areas that bring in a whole load of different signals, and they then compute on them, a little bit like a very fancy transistor. And if the right set of signals come in, then an electrical signal is fired, and then output from that neuron goes to other neurons. So those synapses that Michael already mentioned. So again, a synapse where one neuron plugs into another. So the brain is full of neurons. It's a massive tangle of, beautiful tangle of neurons. And I guess there are probably two really big questions in neuroscience, I would say. One of which is how all these neurons are connected. And then the second is, how does that actually let them work together to solve complex problems like, how do we get to the Wellcome Trust this morning? Or how do we persuade our daughter to leave the house, or whatever it might be that is challenging you on a particular day? So we'd love to solve these problems in the human brain, but the human brain is big from an experimental perspective. So a lot of people use the mouse brain there, which is shown to scale. But as you've already heard, we use the fly brain, which is shown there also to scale. So there are various reasons for that. One is just a question of numbers. So as you've already heard, I actually use the rounded number of 100 billion neurons. So maybe I don't get the cookie or whatever. But actually, it turns out that a group recently used the same methodology to estimate the number of neurons in the fly brain, and they were off by a factor of 2. So I'll wait to see whether the 86 billion stands eventually. And maybe some of us have a few more or a few less anyway. So there's about 1,000-fold fewer neurons in a mouse brain and 1,000-fold again in a fly. So 100,000 neurons, that's sort of a countable number, right? So I know many, many of these neurons. They're my friends these days. And that means that you can do a different kind of science. So the other important thing about flies is they're actually not that stupid. And there's all kinds of stuff which this little animal can do in terms of behavior. In fact, it's a very complex set of behaviors compared to the number of neurons actually in the animal. And that's another reason why it's a good model system for understanding how circuits of neurons control behavior. So I'm going to give you one really simple example of a behavior, but show that there's a wrinkle that you might not realize. And if you take nothing away from this lecture except this, hopefully this will tell you how to swat a fly in future. Okay, so this is an experiment in which there's a virtual fly swat that's approaching. And then this little fly here, she detects this oncoming signal. And watch what she does. So you need to look at the middle of her 3 pairs of legs. Okay. So she moves them, and this is going to let her jump away from the oncoming fly swatter. Okay, so your top tip, if you need to swat a fly in future, is you need to swat where it's going, right? Which is going to be away from where the swatter is coming. So, got it? Yeah. So if it's there, you want to go there. All right. So. We're studying the fly from the perspective of actually having wiring diagrams. And this is really, really important, we think, because the brain is a network of connected neurons. It's like studying a chip. And in fact, some of the technologies we use are exactly the technologies that chip manufacturers use to spy on their competitors. So this idea of trying to obtain a complete wiring diagram of a brain is not new. It's actually due to one of my illustrious predecessors at the LMB, Sidney Brenner. And so he and a small group of colleagues, starting in the early 1970s, decided they wanted to map a whole nervous system. And they did this for the nematode worm C. elegans, only 300 neurons. And the process was basically to salami slice the nematode into several thousand slices, very, very thin, less than 1/100th of a human hair, and then to image them on an electron microscope. And that's because neurons are quite long, can be a meter, right? But synapses are tiny. And so the scale bar is mysteriously, it's up there. So it's 500 nanometers, that white bar. It's less than 1/100th of a human hair. And so that's why we need electron microscopy. And the process is basically you take each slice and you figure out which neuron in one slice that you've cut across matches a corresponding profile in the next slice. And so back in the day, this was done with colored pens by this lady, Eileen Southgate, who apparently loved puzzles in her spare time. And there she is feeling pleased after mapping these 302 neurons and submitting the paper to the Journal of the Royal Society in 1986. And so they called it as a sort of, the mind of a worm was sort of their pet name in the lab, but it actually ended up as the running title of the journal article. Now, this was a really foundational piece of work, but it was also, in all honesty, ahead of its time. And basically nothing much happened in connectomics for 20 years. And then, starting in the mid-2000s, new technology started to come online. And this now means that we can map bigger brains, like my fly, but the approach is essentially a computer-assisted version of what I just showed you. So let's fly into the brain of the fly here. So it's about 1 millimeter across, that purple structure. We're going to look at one electron microscope section, and then we're going to start scrolling through. Each of those circles is one neuron, and we can follow those neurons. Now we can have an AI that actually pulls out the shape of the neuron and then starts to recognize the partner neurons and pull out their shapes too. And if we do that for the whole brain, then we have a connectome. So a connectome, like a genome, is a complete description of the wiring in the brain. You need the shapes of all of the neurons plus their connections, and there we have a connectome. So there is a wrinkle, though, which is that the AI still leaves mistakes that needs fixing by humans. So on the left there is one neuron. It's a sort of tangled tree-like structure. And all the red bits were mistakes that were left behind that shouldn't have been added on by the AI. And the green bits were bits that should have been there, but got left off. And so it took humans, including me, 3 million edits to fix the mistakes left behind by the computers. And that was a big task, so about 30 person years, in order to turn the original image data into something that was usable. Now, there was another bit that was important, though. So, it's amazing what we can do with, a mobile phone in our pocket. But imagine that Google Maps only had satellite images, right? Just the images that you see there. It would be a whole lot less useful. You wouldn't be able to type in and say, I want to go to the Wellcome Trust on Euston Road, right? So the labels on those maps are actually normally the space in which we operate. And so adding labels to all of the neurons in the brain was another key thing that we did during this project. And so this means that our colleagues can go in and let's say they're interested in some of the neurons in the central brain. They can say, okay, let's have those, please. And maybe they're interested just in looking at the neurons on the left-hand side of the brain for some purpose. Let's have those. Maybe they're interested in navigation. So they're going to look at the central complex, which is the navigation center of the fly. Okay, let's have those. Maybe they want to study one particular system within that, the compass system here, which resides in this sort of donut-shaped structure, the ellipsoid body. And maybe then they want to pull out one set of neurons which responds, which are visual compass neurons receiving visual inputs. So they can do that with this interactive map, find the stuff they need, and then figure out how those neurons talk to other neurons. So this is beautiful, I think, at least. And it's also, and it's also pretty complicated. One of the things that we did with this labeling is to take those 139,000 neurons packed into the brain and then group them into cell types. So still quite a few, 8,000 cell types. But each of those cell types is a small group of neurons doing the same thing. And that reduces the set of problems that we have to solve in order to figure out what the brain is doing. Now, it's been a long-term criticism of connectomics that we might be basically looking at snowflakes, right? Essentially, that one brain might be so different from another brain that we're not really going to learn that much by just doing one brain. And connectomes are still expensive to generate. We can't make lots of them yet. And so this is, an example from a colleague. And he says we need to think about the principles of how you make snowflakes. But actually we thought, no, what we actually need to do is collect at least a few snowflakes and see whether they're the same. And so that basically means, can we reliably identify these cell types across brains? So right now, we don't have a whole lot of connectomes, but when we did this comparison, we had sort of 1.5. 3 generously, because each hemisphere of the brain is more or less independent. And so we looked to see whether we could find the same cell types from this new full brain connectome shown in blue to what we had worked on with colleagues at Janelia a few years before, shown in orange, this partial connectome. And indeed we could, and this made us feel a lot better. And just to give you an example, on each side of the brain here, there's an orange cell type on the left, and then its homologous one on the right in blue from a different brain. They've been superimposed, and we can show that they look the same, they have the same connections. And so this feels us much better, makes us feel much better about the utility of these connectomes. So, okay, great. We have a good connectome. What can we do with it? And the truth is, I could spend hours talking to you about this. There are so many answers to this question. There are thousands of researchers around the world using this information now. And I'm going to give you just one example. So some of you may have heard of the work of Ivan Pavlov, who studied a particular form of learning that we now call Pavlovian or conditioning or associative learning. So what he did was to train dogs to recognize that a sound, like a bell, might precede the arrival of some food, okay? And so originally the bell had no meaning for the dogs, but after they'd been paired for a while, the dogs would start salivating before the food arrived. No smell, but they still start salivating when they hear the bell. So flies can do this kind of learning, and it happens in a structure in the middle of the brain called the mushroom body. And there are a few key kinds of cells in there that are important. So one kind are the dopamine neurons. So they bring in punishment or reward. So for example, the green neurons would have been firing when the smell of the food or the process of consuming the food produced a reward. And then there are output neurons here, which can be classified into those that promote approach or avoidance. So whether the animal wants to go towards something or go away from it based on what it's learned. So what we were able to do was connect those neurons with steering neurons that actually let the fly control how it walks. So these steering neurons, there's a pair in particular that you see there. If you turn the one on the left on, then the fly will walk left. If you turn the one on the right on, the fly will walk right. So what we found was that these memory readout neurons connect to those steering neurons with this pattern. So they actually cross to the other side of the brain. And then we needed to do something else. And this was to find out the signs of the connections. So these wiring diagrams are great, but originally, until now, it's not been possible to know whether one neuron was turning another neuron on or actually suppressing its activity, turning it off. And both of those things happen in the brain, and that's critical if you have runaway activity, for example, in epilepsy. So we used, in collaboration with a colleague, Jan Funke at Janelia, a neural network that was trained up to recognize either positive or negative excitatory or inhibitory connections. And we did that for all of the connections in the fly brain. And so with that piece of information, we could then look in a bit more detail at this circuit. And what we could see is that the avoidance output neurons excite the steering neuron on the contralateral side, the opposite side of the brain. And so that means it's going to turn away from something that's activate, a smell, let's say, that's coming in on the left-hand side of the brain here. And conversely, the approach output neurons inhibit those steering neurons, and that means that it's going to steer the other way. Okay, so this is just a very simple way in which we figured out how you can read out a memory and turn it into action. And it's just come straight out of looking at the connectome. So current research. So right now we're working on a complete male brain and nerve cord data set. This is great because now we can look at how the brain controls the motor neurons in the nerve cord and lets the fly, wiggle its legs or flap its wings. And And we can also do comparisons looking at the difference between male and female brains, which is actually quite instructive for a range of behaviors. So in there, we found, for example, on the left is a special pair of steering neurons, which is only present in male brains, but not females. And on the right, in blue at the bottom, is a set of visual neurons that have extra branches that make extra connections only in male flies and not in females. And we think both of these are specializations so that the male can more effectively chase the female during courtship. So there's another behavior which is sexually dimorphic, which we've looked at that you might not know about in the context of the fly. So male flies, compete for mates and resources like food. And if there's not so much of that around and other male flies, then they might do stuff like this. Okay, so this is sort of, the male fly body slamming its rival. And after a few seconds of this, normally one of them will walk away as the loser and the winner gets to, keep the spoils. So we actually have known for some time some neurons in the middle of the brain, the green neurons that are shown on the purple background, that if you turn them on, and that's what's happening in the pair of flies in the left, those neurons are being turned on artificially, and the flies just go at it, even though there's nothing to fight about. The control flies don't. Now, we were able to find the green neurons in the connectome and then look at what information was coming in. And what we found is that actually there's a set of pheromone inputs coming from the legs. So flies can taste with their legs, and they can taste female pheromones with those legs. And basically, this is bringing information to the fighting control neurons that there's something worth fighting about. And so, trying to understand how those control neurons decide when it's time to fight based on the particular set of inputs is actually a really good example of just a decision-making process in its specifics. So, new directions. Flies are great, but maybe there are other animals we should think about. And one animal that's actually relatively close evolutionarily to the fly is this, I think, the yellow fever mosquito, Aedes aegypti. And I think it's reasonable to say that mosquitoes are the most dangerous animal in the world, and that's, of course, because they find humans, let's say one particular human, and they spread disease while getting their blood meals. So how do they find a particular human? So if we imagine we let a mosquito in this room, how would it find Michael or anyone else? So it uses body heat, uses carbon dioxide, and in order to distinguish humans from other animals that might be around, it also, in the case of Aedes, uses particular human odors. And so what we'd like to try and do in a new project with collaborators in Boston is basically to figure out how does the mosquito form a percept of its target human in its brain. And we've already been able to go in and identify, for example, the carbon dioxide neurons within an Aedes mosquito EM volume. And this is great also because when we look at those neurons, we realize actually that they're very similar to the carbon dioxide neurons in the fly. And so we think we're going to be able to reason a lot about the brain architecture from the mosquito, which is still hard to study experimentally by comparing it with the fly, where we've got so many experimental tools. So what about the sort of bigger picture future? So connectomics is really accelerating at the moment. This first worm connectome back in 1986, and it took almost 35 years at least to have another connectome of a whole animal that was even 10 times bigger. But then a year later, we had a connectome 10 times bigger again, more than that. So where are we going? So we'd love to be able to do connectomes for mammalian brains, no question. It's still a big ask for a mouse brain. As I said, over 1,000 times more neurons. And for a human brain, I'm not really prepared to speculate when we might be able to do it because of the scale. To give you an idea, if everyone in the world had a smartphone, and if they got rid of all their selfies and cat videos, and they just filled it with image data from a human brain, the whole world could store one human brain. So it's clear that computational infrastructure needs to move on. But a mouse brain, that's something we can think about in the next 10 years. And there's quite a lot to do. So one of the problems is just the imaging brains, certainly with electron microscopy, which still is what we need to see synapses. is tough. So the state of the art is 1 cubic millimeter, and we're now pushing that to several cubic millimeters. For scale here, there's a mouse brain. There's 500 cubic millimeters in that mouse brain, and there's a fly brain stuck inside it. It's smaller than even just one neuron in that mouse brain. So imaging is tough, but actually, and this is from a report that we was commissioned by Wellcome a couple of years ago now, the biggest problem would actually be fixing the mistakes that the AI leaves behind. And so our estimate right now is that this would be a $20 billion project. I'm not saying it mightn't be worth it, but I haven't yet sort of found the chutzpah to come and ask for it. Yeah. But the good news is that things are getting faster quickly. There's a kind of Moore's law of connectomics right now. And at the current rate, we can imagine that we may have a 1,000-fold speed up, which brings this to a much more manageable $200 million. And so that's basically, where we're skating in some sense. That's where we're going. And that speed up is going to come from better data and better algorithms. And for example, here, to a non-expert, I don't know if you see much difference here, but the images on the right from a new imaging system just off a microscope from our colleagues at Janelia give us significant hope that we can get much lower error rates from the segmentation. So I'd like to finish up then by saying that connectomics provides a key missing ingredient for understanding brains, so the actual wiring diagrams that let brains compute. From early beginnings, actually at the LMB, my own institution, we now have the first connectomes of animals with eyes, legs, and complex behaviors, and they're being used all over the world now to understand how brains actually work. We're working towards mammalian connectomes, and we think this work will have major impacts on basic neuroscience, but also on our understanding of brain disease and even on the development of AI. Thanks again. And this is the big list of people that I need to thank. So big collaborative projects. I just pull out a couple people there. Philip Schlegel at the top, Marta Kostra at the bottom in Cambridge, Jerry Rubin and Stuart Berg at Janelia, Michal Januszewski at Google, David Bock in Vermont. Thank you.
I guess I would like to ask you maybe a little bit about you and kind of what got you into kind of fly brains. What was it first attracted you to fly brains?
Actually, it does kind of link to something I said in the talk, because the same time I was doing that human neuroanatomy class, I was doing rotations in different labs at Stanford. And there was this, as I said, this real disappointment looking at this, this brain and thinking, how are we going to understand that? But I started working in a fly lab at Stanford, where a new technology had just been developed that let you label individual neurons, genetically defined, and put GFP, green fluorescent protein, in them. And then that let us study first the structure of the neurons and then the development and how particular patterns of wiring came about. But also, those neurons were just beautiful, I'll be honest. And I would go into the microscope room and look in the dark and see these glowing green neurons. And I thought, well, I don't know what I'm going to discover, but at least every day I'm doing something aesthetic. So that was actually how I got into fly.
Very good. And actually really important kind of point there about kind of the role of technology and being able to use some of those technologies to view things that may not have been visible before. And I guess it's kind of why for us in discovery research at Wellcome, we think bioimaging is such a kind of a key strand of activity for us. So the work that you've kind of done has relied a lot on collaboration, both kind of international collaboration, but also collaboration across different disciplines. Maybe you'd like to share some of your reflections on that.
Yeah, so I think it's been really important. So in terms of the international collaboration, we've had major collaborations with two groups in the US, one at Janelia Research Campus and the other at Princeton. So I think it was really important for us to be able to work with those people. In one case, the group at Janelia actually imaged the first complete fly brain volume, and they developed new electron microscope technology to do that. The group at Princeton developed a really efficient AI that could segment neurons out of EM images. So our specialism was basically in sort of the neuroanatomy of the fly brain and in merging different data sets together in figuring out how to identify neurons across data sets and various other aspects of what I suppose in bioinformatics happens after you get sequence data and you want to try and turn it into insight. And it was really important to be able to bring these different specialist groups together because actually connectomics was a pretty small field. It's growing now, but it's still not that big. And so we needed that international cooperation aspect and also those different skills. Otherwise, it wouldn't have been possible.
Great. And I guess, you showed us some really interesting ideas and examples of how connectomes can be really useful and helpful in understanding biological or physiological phenomena.
Yeah.
Looking to the future, I guess, you're not predicting when we might be able to get a human connectome, but I suppose looking to the future, what do you think about the possibilities of using connectomes to sort of maybe correct issues or diseases of the brain?
Yeah. So, I think that's clearly a perspective we need to get to. There are a few things that I think could happen quite a bit sooner than a whole human connectome. And if we just extrapolate, it ought to be in about 20 years. But maybe, I'll say that now, even if I don't put it on the slide. But of course, it's possible some transformative technology is just around the corner and we don't know about it yet. In terms of impact for health, I think one of the things that we can think about is basically taking either mouse model systems of disease or pieces of human brain that actually have a problem. For example, a sample from an epileptic patient, maybe after resection, or a sample from someone with a mental health disorder, most likely post-mortem. I think those are things that we could do. And there's a real problem because we don't understand some of these diseases, in terms of having visible pathology. So there are diseases where you look inside the brain and you see a hole, and it's pretty obvious why that brain is not working properly. But there are other diseases where we don't see anything, and we need to look closer. And one of the reasons might be that there's a circuit dysfunction. Some connections are not right. And until we can figure out how to map connections in the human brain, we basically won't know that. So I think that's really where I see that going. So there are lots of interesting comparisons to make to the development of genomics, and we often have those in the back of our minds. The data are not the same, right? So these are 3D image data rather than a linear sequence of bases, but there are definitely similarities. The idea of a reference connectome, absolutely. So people are using this, fly brain connectome to put all kinds of information, and it's not just the information that was actually in this connectome. Sometimes it's other stuff. So, for example, molecular information about the expression of particular receptors in the brain or something like that can now be superimposed on this structural wiring diagram. And then that can help to lead to functional insights about how neurons are talking to one another. So it is, people use Rosetta Stone a lot, right? But it is an integration platform. And it's, sorry, just thinking about the VM. But, it is really the situation that you can have one or two of these connectomes and they can have a huge impact through integration. In terms of, thinking about the mouse and what connectomes you'd want to get, well, hopefully in the UK, we could still have a male and a female connectome. And I think that would be quite helpful. I think, you would need to think about how things change over development. I'm sure you'd start at, some age that was sort of, mature adult, but not old. So there will be important questions to make there. You'll have to think about the strain. None of these, starting now, right? We should have kept the body of these flies and sequenced them, but nobody actually did. That's another thing that we should think about, right? Yeah. So lots of good analogies to consider.
I'm just curious if your work has sparked any interest from philosophers and people who are kind of thinking about what it is to be human and understanding the brain from a molecular and connectomics point of view versus
I haven't really. I did have to give a talk at the Royal Society 3 or 4 years ago, and I did get into some correspondence with a philosopher after that, but he didn't sort of, to be honest, go very far, at least for me. But there was a question that I got asked recently by actually a different group. So a group came from Tsinghua University in China, a group of MBA students. And one of them asked me, so with this connectome stuff, are you going to be able to design smarter babies? I was like, ooh. And then, but then actually, there was something, at least for me, scientifically interesting, which this touched on. So when we compare the two sides of this connectome, we find that they're not identical, okay? So they're very, very similar, but there's a fraction of neurons, sort of fraction of a percent that do something different on each side of the brain. And that's in spite of the fact that this is obviously the same individual. And we think what's happening there is sort of noise in development. It goes right 99.5% of the time, but occasionally there's a little mistake. And we see the same thing when we compare, in fact, this complete brain to the half brain data set I mentioned. And so what we think is that basically every fly is walking around with a slightly different wiring diagram. And in spite of the fact they're all inbred flies from the same genetic strain, they don't develop identically. And I find that a little comforting, because to me it suggests that it doesn't matter how much we engineer our genomes, we're not going to be able to specify our wiring diagrams precisely. I think I like that. So I guess, one important thing, by the way, right, is so far, no connectomic method works in vivo. Okay, so no connectomes until after death right now, I'm afraid. And by the way, there are people thinking about, the brain preservation and, fixing their brains so that they're suitable for connectomics after death, which
you see the Black Mirrors coming.
But in terms of sort of legislative impacts, I suppose, in the short term, I don't think it's going to be connectomics that really drives that, because we can provide wiring diagrams, but we're not telling you how to change the brain. So it's all the other research that comes on top that might possibly do that. But I guess that still doesn't mean it's not our problem at all. I think, one of the things that we do really need to understand simply from a scientific perspective is actually how exactly can we make the relationships between wiring diagrams and brain function in an organism as complex as us. And I don't think we know the answer to that. In a fly, I'm fairly confident that I can engineer some connections and change the behavior of the animal. Whether we'll be able to do that for humans anytime soon, I'm maybe not so confident, even if we have the information. I guess I'm still dodging the legislative question as to how to approach that. But I think we'll probably have quite a lot of time to ponder. So I can actually give one example from our very small number of connectomes. So I showed you this full brain connectome and a half brain, that we worked on previously. And it turns out that when we counted the number of neurons in the memory center, the mushroom body, they were different between the two animals. So in one animal, there were 2,000, in the other, there were 2,500 on each side. When we looked at them more closely, we saw that it was a particular kind of gamma Kenyan cell that is the most numerous memory cell in the brain that was twice as numerous in one fly versus the other. They were the same genetic background. In theory, they'd been handled in the same way, and so it was a bit strange. We looked into it a bit further, and it turns out that what must have happened is that the reference fly connectome is from a fly that was starved as a baby. So basically, the little larvae, maggots to you probably, were probably too numerous in the little food vial, and they competed with each other for food and growth restricted. And it turns out that when that happens, these memory neurons actually increase in number. And so there's a particular developmental plasticity which results in that. Now, why that should be, maybe, it's a bit harder to tell. Maybe, they had a deprived upbringing and therefore they want to, be better at seeking out new food resources and therefore they have more olfactory memory neurons. But it's actually a sort of cool little experiment that was hiding there inside the connectome that shows life experience impacting the brain.
And on that amazing note, thank you so much for enlightening us onto the world of connectomes, stimulating a lot of interesting discussion, and actually thank you so much for making it so accessible and straightforward to follow. So join me in thanking Greg and wishing him all the very best with the work.
And I'm just going to interrupt to say, of course, thank you to you, because Wellcome has funded this work for the last 9 years, essentially the rebirth of Connectomics. And it was very early support, actually, compared with the rest of the field. So we appreciate it.